Hello, I'm Evan, and that's Jay over there. And we're going to be presenting Scikit-Bio. So what is Scikit-Bio? Scikit-Bio is a bioinformatics library that implements data structures, algorithms, and file formats that are used in bioinformatics. We just hit beta as of 8/26 last night, so we're really excited to show you some of the new features we have. Our target users are data scientists with our excellent data munging capabilities, students with our extensive documentation and companion book, An Introduction to Applied Bioinformatics. You can find that at readiab.org, as well as developers for all of the above reasons, as well as our documented API lifecycle. So let's get started. Law's first law says that the first step in developing a new genetic analysis algorithm is to decide how to make the input data file format different from all pre-existing analysis data file formats. We can see evidence of this through about a 5-minute Google search. We currently support these formats. If you have your own format that you need for your research, come by the sprints. We'd love to help you sprint on that. So I/O in bioinformatics is kind of hard, and it's hard for a couple of reasons. One is format redundancy. Many of those formats I just showed you carry essentially the same kind of data. But for whatever reason, in accordance to Law's first law, they changed something, rendering all existing parsers useless. Format ambiguity. Often you don't know what the file format you have actually is. Even with FASTA, one of the most common file formats in bioinformatics, there's no standard extension. You could have your format in .fasta, .fna, .fa, and if you're really unlucky, .txt. And because our data is often in plain text, or these formats are often in plain text, you'll find them compressed so that they don't take up 30 gigs. So Scikit-Bio needs to be able to handle this transparently. So how can we solve this? We can use an I/O registry. And what that'll do is it'll centralize all of the messy logic for reading different sources, figuring out what the file is and if it's compressed, and provide a clean API to both the format implementations and the rest of Scikit-Bio. So here's an example of format redundancy. We have a FASTA file and a FASTQ file. So FASTA and FASTQ hold essentially the same information. The basic difference is FASTA requires a qual file to store quality about the sequence. FASTQ just inlined that and changed all the delimiters in accordance with Law's first law. So we can read this into Scikit-Bio, and we'll see the repr. And so we can see there's some metadata and there's the sequence. And if we compare them, we see they do in fact hold the same information. Sometimes you don't know what your file is. Here we have a mystery file. It's apparently gzipped. Fortunately, Scikit-Bio doesn't really care. It'll sniff that and figure out what it smells like. In this case, it thinks it's an LSMAT file. This is something Chime uses, and it suggests you use a comma as a delimiter. Scikit-Bio can read many different kinds of sources. So here's an example of a NUIC file, which represents a tree. It happens to be gzipped, and it's hosted at a URL. Scikit-Bio will fetch that, turn it into a tree object, and we'll print the ASCII art of it. Okay. Hold on. Is that better? That's probably as much as I can zoom. Okay. We can also read a BZ2 file from a file path, and we'll do the same thing. And so, once again, it reads it. Or, in an educational setting, it can be useful so that the students can see what the data actually is. So NUIC kind of looks like this. So it will also read a list of lines. And we get the same tree. So in accordance with Law's first law, let's create a format for this presentation. We'll call it Yossef, yet another sequence format. We invented this on the plane flight over, and it looks like this. So we see we've got some basic metadata. We've got ID, location, description, and sequence. Our killer feature here is location. FASTA doesn't have location. We can tell Scikit-Bio about this in a fairly straightforward way. First, we create the format for Scikit-Bio. So we'll create a Yossef object that represents the format. Then we can associate a sniffer, and its job will be to determine, does this look like a Yossef file? We've made it easy on ourselves by using a comment that says Yossef at the top of the file, so we can very easily detect is this Yossef. You'll notice that it doesn't have to worry about compression or encoding here because that's handled transparently by the I/O registry. So you only have to worry about the business logic. We'll also associate a reader with DNA. So this will turn a file handle into a DNA sequence. So we'll execute that, and now Scikit-Bio knows what Yossef is. We can prove that by showing this. And so 2 seconds ago, DNA had no concept of Yossef. Now it does. We see description, ID, and location. Obviously, that was a terrible idea. None of the existing bioinformatics tools work with Yossef. So we'll convert it back to FASTA so that we can actually do something. So we'll write that back out as not Yossef.fna. Of course, we could have chosen .fasta. And so we'll see we've got our ID and description and sequence. We did lose location because FASTA doesn't know what that is. So hopefully you can see how we can normalize all of these different input formats into a consistent object that you can manipulate. But can you really trust that object? Jay is going to talk about our API lifecycle now.
So as Evan just mentioned, we just hit beta last night. And you might be wondering, should you be using Scikit-Bio for your own work or in production? To help alleviate some concerns with the relative newness of the project and its beta status, we've put together a diagram illustrating the API lifecycle, and we're committed to applying this lifecycle to all of the public-facing API in Scikit-Bio from this beta release and moving forward. So when new functionality goes into Scikit-Bio, it's typically going to enter an experimental state for a number of releases until it's been vetted and it's been used and proven to have a reasonable API that fits into the rest of the Scikit-Bio ecosystem. From there, we would transition that API to stable, and ideally that API would stay in Scikit-Bio for the indefinite future. But if we do decide at some point to change that API or to remove it, it would transition into a deprecated phase. And if you were using that functionality, you'll get deprecation warnings saying, hey, this is going to change or be removed at this point in the future. And eventually, like 2 or more minor releases in the future, it'll be removed from the package. It's a similar process for experimental functionality. If it doesn't make it into stable, it would also be deprecated and then removed on an accelerated time schedule. So we hope that this will make it clear what's stable and what isn't, so that you have some guarantees about what's likely to change and what isn't in Scikit-Bio as we progress through more and more beta releases. Just to demo what this looks like, we present this information in the doc strings that you'd see on our website or if you called help on a function. And so we implement this with decorators. We have like a stable decorator, an experimental decorator, and deprecated decorator. So let's say that we define a really simple function here that adds 2 numbers, and we're really sure it's right, so we're going to mark it stable as of version 040. If you look at the doc string here, you'll notice it's in NumPy doc format, but there is no indication of the stability of this API yet. If we execute this and execute this decorator, and then call help, you can see the same doc string is here, and it's had a line added indicating the state of this API, indicating that it's stable as of version 040. And likewise with experimental and deprecated, this information would be displayed in a similar way. Currently, the I/O functionality that Evan just demoed is marked stable, and so are the sequence objects in Scikit-Bio, which I'm going to demo right now. So we've seen that Scikit-Bio is pretty awesome for doing I/O. We haven't really seen how it fits into the rest of the SciPy ecosystem, which is an important part of being a Scikit. So I'm just going to go through a few API examples to sort of show how it can interoperate with NumPy and Pandas and Scikit-learn and so on. Let's say we have a DNA sequence here that has some casing. What Scikit-Bio is going to do is internally it's going to normalize that casing into uppercase to be valid IUPAC. But unfortunately in bioinformatics, case is often used to represent some sort of other information like introns versus exons. And so we also provide an easy way to extract that information and store it as metadata on the sequence object so that it's not lost when the casing gets converted to upper. So let's go ahead and just run this and see what the sequence looks like. So we can see we have DNA with some positional metadata. We have a column called exon that's basically a Boolean vector storing that casing that was in the original sequence. And if we look at the actual sequence that's stored in memory, you can see it's been normalized into valid IUPAC characters. Sequences, internally, they're backed by NumPy, and you can get at the underlying NumPy array by calling .values, which is kind of similar to how you would get at the NumPy array of a pandas data frame. So you can see we get the NumPy character vector back. The positional metadata is stored as a pandas data frame. So there's that exon column that we created when the sequence was instantiated. This is a Boolean vector that we can use to actually slice into the sequence to remove the introns and get a sequence back that only has the exons. So we can just slice like so with our positional metadata. And the sequence you get back, you'll notice, is shorter than the one before. It's had all of the false positions removed. So just to wrap things up, we're going to show a little more complicated example of how you can use Scikit-Bio and Scikit-learn to build a taxonomy classifier. And so real briefly, what a taxonomy classifier does is tries to predict what organism an environmental sequence likely came from. And so you can train a classifier on sequences where you already know the taxonomy or what organism the sequences came from. And so we're going to just real quickly show how you can do that with Scikit-Bio and Scikit-learn. So first, I'm just going to define a couple data paths here to the reference sequences. So the reference sequences, we know what their taxonomy is. And we're going to need to do a little bit of pre-processing on the sequences to extract out the region of interest that we want to actually train the classifier on, because these sequences are very, very long. We're going to do that by defining two short sequences using Scikit-Bio's DNA object. And then I wrote a little helper function here to basically create a regular expression from a sequence. And we need to do that because there are degenerate characters in those primer sequences I just created. So executing that, we now have a regular expression. And we can use Scikit-Bio's I/O framework to read in this FASTA file of our reference sequences. And then we can use find with regex, which is a method on the DNA object, to perform these regular expression searches, ignoring gaps in the sequence. And we can record what the start and stop positions are. We're basically finding the region that we want to slice these sequences to. So if we run this, you can see we end up with a slice with start and stop. Next, we can take another pass through our reference sequences. Now that we know those positions we need to slice by, we can use I/O again. And for each sequence, we're going to slice the region that we want, degap it using a degap method. And then we're going to use a method called kmer frequencies to split this sequence up into words of length 8, or kmers. And we're going to actually use those as the features to train the classifier on. So now we have a dictionary mapping kmers, or mapping words to their frequencies or their counts. And so the next step is to import some functionality from Scikit-learn that'll convert that dictionary into a vector that it can consume. Next, we're going to load in the known labels, or the known taxonomy. So this is just some code just to parse that file and get it into a structure that Scikit-learn can consume. We're going to do feature selection to throw out features or words that aren't informative. And then take our data set and split it into a training data set and a test data set so we can do cross-validation. Finally, we're ready to instantiate an SVC, or a support vector classifier. And we'll pass in our training data and see how well it predicts on the test data. And we can visualize these results as a confusion matrix and an F-score. Let me zoom in. Oops. So it's maybe a little hard to read. On the x-axis here, we have predicted taxonomy. And on the y-axis, we have known taxonomy. So what we're looking for here is ideally a solid diagonal. And we're hoping for an F-score that's close to 1. So the demo I just ran is on a very small subset of the full set of reference sequences we have. And we ran this last night and on the full data set, and we got an F-score of 0.95. So it appeared to do much better using the full data set, but it just takes too long to run for the demo. So hopefully this demo kind of illustrates how you can use Scikit-bio's API to do your data munging, your preprocessing, and then feed that directly into something like Scikit-learn or something else in the SciPy stack to implement something fairly complicated like a taxonomy assigner in very few lines of code. Zoom out. With that, we'd be happy to take any questions. So as far as like direct integration, we haven't really made any plans or progress towards that. Scikit-bio has, the way I see it, a different goal than BioPython. BioPython kind of fits into like the other BioStar projects like BioJava, BioPerl, and so on as far as its API and design. And so we really want Scikit-bio to fit into the SciPy ecosystem and the SciPy stack. And so we're making very deliberate decisions to, have things backed by NumPy and, SciPy data structures, and to work with things like Boolean vectors and so on. And you're probably not going to get that with BioPython because they just have like a different take on designing this object model and this API.
So some of the things that are going to drive Scikit-bio's development is basically a redesign of QIIME 2, which does kind of a full bioinformatics suite, at least from a microbial ecology perspective. Obviously, we want to support all kinds of bioinformatic fields. And so the next things on our list are probably alignment, sequence collections, and trees. And we need to figure out kind of how those work. We're probably going to chunk through those experimental sub-packages and bring them up to stable.
How do you decide to package from experimental to stable version?
We'll do that kind of after it's been vetted for a while. It seems to have a stable API, or it seems to have an API that it acts like a stable API. So we haven't had to like tinker with it. It's being generally used, and it's pretty much ready to go. So it's subjective. Yeah, it's subjective.
Yeah.